Hi everybody, let me tell you in detail about the CAMP test. C-A-M-P, CAMP test. CAMP stands for the name of three scientists who described it in 1944. That is Christie, Atkins and Munch-Peterson. This test was developed initially for the detection of group B streptococci that were responsible for producing mastitis in cattle. CAMP test can be done only with beta hemolysin producing strains of Staphylococcus aureus. An example of such a strain is the Placent strain, that is ATCC 25923. Let me tell you about the procedure of doing CAMP test. Take a sheep blood agar plate and streak the Placent strain as a straight line in the middle of the plate. Now take the test strain of group B streptococci and streak it perpendicular to the Placent strain, but the two should not touch each other. There should be a gap of 5 to 6 mm between the two. Positive and negative controls can be streaked on the same plate perpendicular to the Placent strain. The streptococcal streak should be about 3 to 4 centimeters long. Label each streak on the back of the plate. Streptococcus agalactiae ATCC 12386 is the positive control for CAMP test, and Streptococcus pyogenes ATCC 19615 is the negative control. After overnight incubation, we see an enhanced area of hemolysis at the area where the Staphylococcus and group B Streptococcus intersect. The hemolysis appears like an arrowhead. If hemolysis does not appear enhanced, the inoculated plate can be refrigerated. This is because beta hemolysins show a hot-cold phenomenon. The beta hemolysin produced by Staphylococcus aureus is a sphingomyelinase. It is an extracellular protein. The hemolytic property occurs only in the presence of magnesium ions, and it acts only on cells with sphingomyelin and lysophosphatidyl-choline. Sheep blood is rich in both sphingomyelin and lysophosphatidyl-choline, hence it is ideal for CAMP test. The beta hemolysin shows a hot-cold phenomenon. The hemolysis starts at 37 degrees Celsius but gets enhanced on chilling. Initial disruption of cohesive forces within the cell membrane occurs first, and when the temperature is lowered, phase separation within cell membrane occurs, leading to lysis of the blood cells. The beta hemolysin produces synergistic hemolysis when it combines with CAMP factor, which is an extracellular protein seen in group B streptococci. The principle behind CAMP test is that first, sphingomyelinase produced by Staphylococcus aureus hydrolyzes the sphingomyelin of sheep RBCs and produce a product called ceramide. The CAMP factor of group B streptococci acts on ceramide and immediately lyses the RBCs. A similar type of CAMP test can be done with Listeria ivanovi and Rhodococcus equi instead of Placent strain and group B streptococci. CAMP test is also positive for Listeria monocytogenes, some strains of Vibrio cholerae, Rhodococcus equi, Listeria siligeri, Listeria ivanovi, Pastorella hemolytica, Streptococcus uberis, group G streptococci, Aeromonas species, Actinobacillus pleuropneumoniae, Actinomyces neoe, Propionibacterium granulosum, and some species of Corynebacterium like Corynebacterium imitans, Corynebacterium striatum, Corynebacterium affermentans, and Corynebacterium glucuronolyticum. Another strain of Staphylococcus aureus, that is ATCC 49444, was used for CAMP test, but molecular studies have found that this strain was not actually Staphylococcus aureus. It was another organism which was named Staphylococcus pseudintermedius. So Staphylococcus pseudintermedius is another example of coagulase positive Staphylococcus. All the strains of this Staphylococci can be used for CAMP test. Now let me tell you about reverse CAMP test. This is a test which was described by Hansen in 1980. Here we have to take the sheep blood agar and streak a straight line with Clostridium perfringens, then streak group B streptococci perpendicular to it. Incubate them anaerobically and an enhanced hemolysis occurs at the junction which has a bow tie pattern. It is also called arrowhead appearance, but if you look carefully, it is more like a bow tie and hence it is called bow tie pattern in some textbooks. Alpha toxin produced by Clostridium perfringens, which is a lecithinase, also has got a synergistic hemolytic property with CAM factor produced by group B streptococci. When we talk of enhanced hemolysis shown by organisms in CAM test, generally we describe it as arrowhead appearance, but even this has variations in their presentations. Group B streptococci gives the typical arrowhead appearance, but if you see Clostridium perfringens, they give a bow tie appearance. Again, if you see the CAM test produced by Listeria monocytogenes, it gives more of a rectangular shape of hemolysis. Another method of doing CAM test is the CAM disc method. Paper discs which are impregnated with beta hemolysin of staphylococci are available. The disc is placed about 5 mm near a streak of group B streptococci which is made on sheep blood agar. The plate is incubated at 35 to 37 degrees Celsius overnight. We see a lunar shaped clear zone appearing around the disc. Another method of doing CAM test is the CAM spot test. For doing this test, we need CAM spot test reagent which is made by the beta hemolysin of Staphylococcus aureus. First, the group B streptococci are streaked on sheep blood agar and allow incubation for at least 18 hours but not more than 48 hours. With a pipette, place 1 drop of spot test reagent on an isolated colony. Close the plate and incubate it in the presence of 5 to 10% carbon dioxide for 30 minutes. The plate has to be kept with the right side up so that the reagent does not flow away. Observe using transmitted light. A circle of enhanced hemolysis will be seen next to the colony. If the test is negative, you have to re-incubate for another 30 minutes or refrigerate it. Even after that, if there is no enhancement of hemolysis, then it is considered as CAM test negative. A special mention that has to be made is that a very small percent of Streptococcus pyogenes, that is group A streptococci, also give positive CAM test. In such cases, for identifying the organisms with biochemical reactions alone, we have to do the PYR test, that is the pyrrolidonyl beta-naphthylamide test, to differentiate group A, which is PYR positive, from group B, which is PYR negative. We can also do the bacitracin sensitivity test. Group A is sensitive while group B is resistant. So that's all about CAM test, which is done mostly to detect group B streptococci. Thank you.